Yep. Oh, cool. Okay. Let's do this one.
Yeah.
There's a song.
So we meet.
It's on YouTube. I can, yeah. But go down there.
You're forced to sit next to me. You didn't know you liked me. Or you
It's such a struggle.
Oh, I'm so sorry for you, short person.
This was jumping. I would end up killing this person.
There's another
Oh, yay! I'm so excited to see this. Bring me a king. I hate how this I was like, well, like, how did it turn out? What did I do incorrect? Nothing but clarifying.
So as I'm going to get started here, this is our final 2 talks of the afternoon. Also, immediately after the talks, we'd like to get a group photo with all students. So after the talks, please head out front next to the voice consonant student sign, and yes, Marissa probably will be taking a picture of all of us, or all the students participating in the summer program. But more importantly, I want to introduce our next speaker. This is Jordan Zonner, who is at a Bugabut's lab, and more specifically working with Matt here on CRISPR-Cas9 editing plants, and we'll hear more details about that.
Thank you very much for introducing me.
Hi, I'm Jordan Zonner. I am a biology major with a minor in software development at a small liberal arts school in Crete, Nebraska called Doane University. And this summer I had the lovely opportunity of working in the plant transformation facility here on Cornell's campus with Dr. Matthew Lowen. And together we were testing the use of long single-stranded DNA template for a means of editing with CRISPR-Cas9 in plants. Okay, so what is CRISPR-Cas9? Well, it's a response that bacteria have used against viruses for many years. And so how it's done is there's a single guide RNA, it's represented in this red box, that guides the Cas9 nuclease to a specific 20 nucleotide spot in the target DNA. And once it can recognize this 20 nucleotide specific spot, it also needs to recognize something called a PAM sequence, which is another 3 specific nucleotide spot in the DNA. And from there, it can actually start using these scissor-like endonucleases to make a double-stranded break in the target DNA. And so once that happens, these endogenous repair systems begin to happen too. And there's a couple of different ones that are perfectly performed. One on the left right here is non-homologous end joining. And this one is more prone to having mutations occur. So the one that we want to look into more is the homologous recombination, which is shown on the right. And the homologous recombination is the use of a sister chromatid as a template to repair the broken DNA. So scientists have taken advantage of this system by providing a donor template with a sequence of interest incorporated in between the 2 homologous arms so that the repair enzymes can recognize those 2 arms, incorporate that sequence of interest into the genome. And this is, oh, sorry about that. Okay, so there are some methods for CRISPR-Cas9 editing in plants, and most of them have been transgenic. So with that, you can see right here, this construct has the Cas9 encoding, the gene encoding Cas9 right here in yellow, the gene encoding a single guide RNA right here in blue, and a selectable marker, which could be an herbicide or an antibiotic marker that you use to see if your edits are there from the Cas9. Also, another way, and so when you do this, you're performing a deletion of the gene of choice that you want to do. So with that, the guide RNA leads the Cas9 to a specific spot, cleaves the 2 spots in the DNA, and removes that piece of DNA. You can now actually do another way too, by providing the same construct, but except adding those 2 homologous arms that I was talking about before, which is also right here, the sequence of interest that scientists have used, providing that to the DNA, and having that be inserted into the genome, which is still transgenic. This is all a transgene. And this is used for swapping or inserting DNA, or otherwise called an allelic swap. So there are some also non-transgenic ways of editing too, if you guys want to look into that. And with that, you can use protoplasts. So protoplasts first are isolated from plants, and then you can digest their cell wall, leaving the cell membrane. And with enzymes, you can make the cell membrane porous so that exogenous material can get into the protoplast. And from there, you can regenerate full plants. You can do this by providing the Cas9 protein, the guide RNAs, to make that deletion, like I was saying before. So you can transfect these 2 into the protoplast, see your edits by PCR. And the same thing can be done with providing a donor template. So you provide a Cas9 protein, the guide RNAs, and a donor template. Now, there's another method too that people have looked into, which is also non-transgenic, and it is using the same thing, but it's using a gene gun. So some of you may have heard of a gene gun. Fun fact, it was actually founded on Cornell's campus, so that's pretty awesome. And you can do the same thing, except this time you are using the Cas9 protein, the guide RNAs, and you're coating them onto gold particles or tungsten particles to bombard plant cells with and get that exogenous material into the cell that you want. And you can do the same thing again by providing a donor template. So you have a couple different options when you want to provide a donor template to your repair enzymes to get incorporated into the genome. Ones that have been studied are right here. So you have a double-stranded DNA template, a short single-stranded DNA template, and a long single-stranded DNA template. So the double-stranded DNA template has been found to be transgenic in both plants and animals. And the short single-stranded DNA template has been found to be non-transgenic and also very efficient with plants. Now, recently, there's been studies about long single-stranded DNA template, which has been found to be non-transgenic, providing non-transgenic edits in animals. So that leaves a lack of investigation in the long single-stranded DNA template in plants, which is why we want to look into this. So the questions that we are asking this summer are, can these two non-transgenic methods, are they successful, the protoplast and the biolistics? So let's test them out. And also the second question we want to ask is, does the long single-stranded DNA template actually, can it provide a transgene-free way of editing with CRISPR-Cas9 in plants in comparison to the double-stranded DNA template as our control? And so the next is, the thing that we want to do too, whenever we get this single-stranded DNA template, is we want to modify the ends of the template so that plant nucleases that are can't actually modify and edit the single-stranded DNA template. So it makes your gene editing efficiency greater. So what is the significance of all of this? You guys are probably wondering. Well, most of you have probably heard about all these different moves with wanting to get away from transgenic editing and more of the non-transgenic editing. And so if we can solidify these methods, then they can be applied to a lot of different plant systems, other organism systems. We just want to make it so that we can make stronger staple crops and that it can be applied to any type of plant. So we are going to test these methods with a gene called ART1, which is a gene that is found in rice plants that codes for aluminum tolerance. And this gene has been well studied in other labs like the Susan McCooch lab. And so we decided to go with this because we have access to a lot of information on the ART1 gene. But also, we want to test this deletion and the allelic swap. So, Nepombar is tolerant of aluminum, so we want to look into deleting that with the Cas9 and the guide RNAs so that we can see a phenotype of sensitivity to aluminum. And then also IR64 is sensitive to aluminum. That's what we want to do, is provide that allelic swap. Does our method actually work? Can we cut out the single-stranded DNA template, provide it to the repair enzymes, and get it incorporated into the genome, so that we can see the genome of the Nepombar cultivar, so that we can see a sensitivity phenotype. So this summer I had a lot of time spent with the gene gun. A lot of the methods that we were testing were with the biolistics, the particle bombardment, and we saw a lot of, we had a lot of love, and we also had a lot of different things that we wanted to try again with the gene gun. Love-hate relationship there. So with our bombardment conditions, we do want to test them out first, make them optimal. So the first thing that we decided to do is provide a double-stranded DNA template with that, and have some GFP also on that plasmid. So it's a plasmid that we are coating onto gold particles, which they actually don't look that sparkly as depicted in this image, but I wish they did. You guys will see a picture of them soon. So once you coat them onto the gold particles, then you can go ahead and go forth with the bombardment through the gene gun. And then with that, the first type of plant cell that we wanted to choose, that we chose, was plant callus, which is a type of cell that is an unorganized cell that many plants actually can generate. And from these cells, you can regenerate plants, which is why we wanted to choose them. But when we noticed that we bombarded when we bombarded with the GFP, we noticed that it was probably because we didn't see an expression of the GFP, probably because the cells were too thick. So we decided to go with a different cell from Nepombar, which was the embryos. And so right here, you can see me extracting a Nepombar embryo. And I wanted to show you guys this because maybe some of you don't know how small they actually are. But it's important that we get those isolated because they're way less thick than the callus. So it should be a lot easier to get the exogenous material into the plant cell. So whenever we decided that the GFP was not going to be that effective, we decided to use a different type of fluorescence, which was GUS, and a lot of you have heard about fluorescence blue. So right here, you'll see the Nepombar embryos, and they're coated with the gold particles. And Matthew and I were kind of looking at them, we're like, kind of looks a little bit like cinnamon on a donut. So we were just having that little running joke in the lab. It made things a lot more fun. And actually, it made it even more fun when we saw expression of GUS on the embryos. But as you guys can see, not all of the embryos were expressing Gus, so there's still more conditioning that we need to optimize. Okay, so once we can get those parameters optimized with the gene gun, we can go forth with a real bombardment, which is with the Cas9 protein and the guide RNAs, to perform the deletion of the ART1 gene in Nippon-bar rice. And so the way you would do that is provide the DNA, code it onto these old particles, which I don't know that sparkly, bombard with the gene gun, express and get the edit in the chromosome. The awesome part about Cas9 and guide RNAs is that they will be degraded afterwards, so they won't make those edits elsewhere in the genome. And then from there, you can induce callus by just putting them on some callus infection media. From there, they will regenerate full plants, and then from this point on, you can do PCR. So obviously, you guys can see in the timeline, I won't be here for some of these experiments, but we still are optimizing these parameters so that we can run a full bombardment with that, with the Cas9. So, like I was saying, you would do the same thing, and you would do it with our long single-stranded DNA template, and you could do the same exact methods with this. And that is, again, to perform the allelic swap. Again, we would see a phenotype of sensitivity to aluminum and Nippon-bar. And then also, we want to use the double-stranded DNA template as a control so that we can see, okay, the long single-stranded DNA template is actually providing, or is actually being incorporated, and it's not making edits elsewhere in the genome. Okay, so we haven't, we just started doing the methods for the protoplast. So what you do with the protoplast is you isolate the cells from the rice plant, which is our Nippon-bar, and then you can grow them up in some media, allow them to, or you digest their cell wall, like I was saying, leaving them with the cell membrane, provide them with some nutrient-rich media, or otherwise called a nurse culture, and then you allow them to regenerate and form their cell walls, cells divide, see that they're actually growing and happy, and then you can, once you use enzymes to make the cell membrane porous, then you can transinfect with the Cas9 guide RNAs. From there, you can regenerate the full plants from the callus, just like we did with the embryos, and regenerate full plants. From there, you do PCR to see your edits. Okay, so future directions. Like I said, currently we are still working on fine-tuning those bombardment conditions, and with that, we also need to start synthesizing our guide RNAs. So we bought a kit the other day that allows us to synthesize the guide RNAs at our the plant transformation facility. So that's something that we're going to be working on, I think, today. And then after this, we can, once we can get those all synthesized, we can bombard with the Cas9, the guide RNAs, perform a deletion, and then afterwards we can go forth with, once you can see the deletion by PCR, you want to go forth with the allelic swap by providing the long single-stranded DNA template. And now we're also continuing to let the protoplast grow and thrive. So with that, I just want to thank everyone in this picture. I do want to thank Matthew Woman for taking me in. This is my first real wet lab experience, so it was a lot of different techniques that I learned, but they're invaluable to me. I will always know and remember this experience. Thank you, Delaney, so much for coordinating, and Dr. George Jander, too. Thank you very much. Erin Doyle is my professor that I work with at Doane University, and she also helped send me and another intern here, and it's been a great experience, and thank you very much.
Any questions for Zoe?
I was just wondering if you could explain again why the single-stranded DNA was non-transgenic.
Okay, so the single-stranded DNA template is non-transgenic because once it's inserted into the genome, it doesn't have the risk of random insertion elsewhere like the double-stranded DNA template. The reason why I don't have an answer for right now, so but just know that double-stranded DNA template does have that risk of inserting random elsewhere in the genome. Yes.
How long is a short versus a long strand? Like, what's the differentiation to that?
Well, so the long single-stranded DNA template is about 1.5 to 3.0 kilobase pairs in comparison to a short single-stranded DNA template, but I'm actually not sure how long those are exactly, but
Okay, so yours are maybe.
Yep, okay, yes.
Your cycle time between parameters, how long does it take you to run these?
So all in all, you have to use the embryos and the seeds that day. So you husk them that day, sterilize them, and then the bombardment through the gene gun probably takes about an hour, but all in all, I would say it probably takes maybe like 6 hours to coat the gold particles. Run a gel saying that the gold particles are actually coated with DNA, and then go forth with your bombardment.
Okay, so we should probably move on. So our next speaker is Zak Stevenson, who's also in the Buckingham Lab. And then working together with Andrew Reed and Sylvie Brown. And
Have fun doing this.
they were telling us about Xanthomonas infection in rice.
Thank you, Dr. Jander. Yes, my name is Zak Stevenson, and I'll talk to you about mobilizing pattern recognition receptor FLS3 to increase rice resistance to Xanthomonas oryzae. To give you a little background on the importance of this research is that rice is a staple food crop for 50% of the world. And it is predicted that the global population will soon reach 10 billion food by 2050. And so at current food production rates, this will not be able to sustain the growing population. So this allows researchers to investigate new ways in order to increase the crop yield. Two ways of doing this is through disease-resistant rice or creating tolerant rice like cold or drought tolerance. My focus today will be on disease resistance, specifically to Xanthomonas oryzae, or XO, and two pathways within this. The first one, Xanthomonas oryzae PV oryzae, or XOO, causes bacterial blight, which is where it infects through the vascular or the xylem of the rice leaf and then works and then infects its way down. And the second is Xanthomonas oryzae PV orisicula, or XOC, which causes bacterial leaf streak. And this is where it can get into the wounds or the stomata of the rice leaf and then affects north and south on the leaf. And with this one, you really see an exudate that is in field conditions that if there's wind or rain, this exudate is able to blow around, and that's how it's able to infect other leaves or other plants. Oops, sorry. Now, how is it that the rice actually combats this pathogen? Well, first, it has a pattern recognition receptor, or PRR, that's located in the cell membrane. It recognizes a pathogen-associated molecular pattern, or PAMP, that's within the bacteria on the flagellum, which is the tail of bacteria. And with this recognition, it triggers PAMP trigger immunity, or the first line of defense, and that creates an immune response. A well-known example of this is the flagellum sensing receptor 2, located in rice, that recognizes the flagellum 22 epitope in the Xanthomonas oryzae. With this recognition pair, that activates your MAP kinase signaling and an increase of reactive oxygen species and an increase of the pathogenicity-related gene expression. But there's a kicker. The XO clade 22 evades recognition to the FLS2. So where do we go? Well, it was on previous research done by the Martin Lab. There was found a second flagellum sensing receptor, or PRR, called FLS3. And so it works very similar to the FLS2 in flag 22, but the FLS3 recognizes a second epitope on P. serendae bacteria. And so now we want to figure out, oh, is this conserved in the pathogen XO? And so the sequences were blasted, and we did find that flag 228 is conserved in XO. So the FLS3 gene was cloned and then transformed into rice, where when I arrived this summer, I had my transgenic FLS3 rice, or my T1 plants that I planted, and I allowed me to create the hypothesis. FLS3 recognition of XO, flag 228, in rice will influence the resistance to XO. also at the beginning of summer, we had 3 aims which would accomplish our hypothesis or to test our hypothesis. The first one being the presence and expression of FLS3. The second is FLS3 mediated activation of PTI. And the third is to check for the FLS3 influence resistance to XO and XOC. First, I need to genotype my plants. So I need to find out if the FLS3 was present in these plants. And so I do this by extracting gDNA or genomic DNA. And by PCR, I design primers that would amplify 300 base pair of the FLS3. And as you can see, the white band is a positive FLS3 presence. So once I finish my genotyping, I know which plants have the presence or my gene, FLS3, but I don't know the number of copies. So now, using a qPCR of my transgenic plant gDNA, I found that I had zero copies. I have 7 plants with 0 copies, 23 plants with 1 copy of my FLS3 gene, and then 12 plants with 2 copies of the FLS3 gene. I genotyped and found the zygosity of my plants, and now I want to know if there's actually FLS3 transcript being made. In order to do this, I extracted the RNA in order to perform an RT-PCR. And using the same primers, I amplified the 300 base pair region. And my negatives, and I like to say all my negatives from here on out will be the no FLS3 presence. And so the first 3 lanes will be the homozygous, or just a sample of the homozygous plants. And then lanes 4, 5, and 6 will be the heterozygous plants. And so as you can see, it does go along with my genotyping. The negatives do not have the presence or the transcript being made of FLS3. And then lanes 1 through 6 have transcript being made of FLS3. Now that I know there's transcript being made, I want to find out if there's protein expression of FLS3. So I probed a Western blot with an antibody that recognizes the ACV5 tag that should be on FLS3. And thank you to the Harrison Lab for allowing me to borrow their ACV5. And so with this, you have my negative controls, and then the positives, 3 to 10. But I want to see at 110 kilodaltons, the bands, but you do not. And so we do not see protein expression. This could be because there might be too low of levels of FLS3, so we're not going to be able to see it. Or maybe it's not being expressed. But we did have an idea, which is to stabilize FLS3. And in order to do this, I infiltrated rice, and then at time increments of 5, 10, and then 1 day, I would collect the tissue. I would then use a Western in hopes that this would actually allow, or I would hope that it would bind the FLS3 to the flag 228, and this would then stabilize it. But as you see, at 110 kilodaltons, I still do not have protein expression. But in the previous research, this is actually something that happens often because FLS3 degrades so quickly. Sometimes you just don't see it, but it is still functional. So we keep going. And so this leads us to aim 2. It's an FLS3 mediated activation of PTI. And so I was not able to get to raw production, unfortunately, but there'll be more on that later. And so I go to my pathogenicity-related gene expression. And so this, I wanted to see if there's a change in the transcript of the PR genes. And so I designed primers based on different PR genes and that I found in literature, and I used a QRT-PCR to determine the fold change. There was, as you can see on the proxies, there was some upregulation compared to my negative infiltrated rice. But this is still preliminary results, and so we would need to do more replicates, also test more PR genes in order to confidently say this is an upregulation. Okay, so then I checked for FLS3 influence resistance to XO and XOC. And I infiltrated my rice by syringe infiltration with XOC. And this is by, so when you syringe, you just push the bacteria in through the stomata, or even if those are one of those actual conditions. And as you see, after 14 days, I measured the lesion length, and there was no significant difference. So then I checked my XOO infiltrations. This is done by dipping scissors into bacteria and then clipping the tops of the leaves because that's how it gets into the vascular system. As you see, I measured the lesion lengths for the spread, and there's still no significant difference between my positive and my negative. This comes to my conclusion is we do have a presence of FLS3. There is transcription of my FLS3 as well. And preliminary PR genes are, that shows upregulation, but we cannot confidently say until we do our replicates that it is upregulating all genes. But it does not show influence on resistance. And the protein expression may be too low, or it may just not be functional. But that will come on where do we go from here. As before I said, we were not able to determine our ROS production. Well, this is a hallmark for the FLS3 action, and this would show if the FLS3 is functioning. So I would definitely need to perform this experiment in order to actually see what is going on. Another thing I can do is alter the conditions of my bacterial inoculations. So during my experience, I did not have a difference in lesion length, but this does not, it's not indicative of the field conditions. So maybe painting on the bacteria would be kind of the spread through the rain or wind, and it's not just forced infiltration. Also, as you can see, you've seen this figure 3 times, and if you notice, there's this new FLSX that was found. And this is preferable that we might not be able to see FLS3 doing anything to the plant, because this FLSX could be something very similar to the FLS3 epitope, but it's recognizing a different epitope, but it could be functioning similarly. So more research on this is needed because it also has not been characterized on what epitope this is actually recognizing. So with that, I'd like to acknowledge Adam Bogdanoff for allowing me in his lab this summer, Zoe Burrow for being my head mentor on this project, but also Andrew Reed for being my other mentor. The Martin Lab for having Project Premise, Aaron Doyle for allowing me here this summer, and then BTI, Delaney, and Dr. Jander, and then the funding from NSF. But also, I'd like to give special thanks to the entire lab because they made this a wonderful, fun, fulfilling summer, including little fluffy dog. But thank you.
Do you know if there's some way for maybe like plants to cut off their xylem so that you can't spread bacteria? Like with local recycling?
I do not know that. Sure, they can cut their xylem. That, yeah, that's a good question. But yeah, I would have to do the literature research on that.
So did I see that correctly, that the FLS3 was from tomato and was inserted into rice?
Yes, so it was, yeah, cloned and then transformed into rice.
I remember in your Western blot picture, you had taken tissue from the plants at 5 minutes, 10 minutes in a day, and it changed so quickly. I was wondering why you would still include the option of tissue collection after an entire day.
And so that's a great question. And so we wanted to see, so it's so quick, the endocytosis where it does degrade back into the membrane once it recognizes. And so at 5 and 10 minutes, I would maybe see, oh yes, it's stabilized. And then that one day would definitely tell us it does degrade quickly and there is no expression after that. And so it's just kind of to confirm the previous, that we weren't getting any bans on the previous Western blot before that one.
Okay, if there's no more questions, thank you very much.
She's a break. She's currently studying FLS3. She's like, she's like, oh my god, I didn't realize anyone else was there. She came, she was in there, just like reading all this stuff. I wonder is, so they put the transgenic FLS3 in, but I wonder if there are other genes that need to be present to incorporate that sort of recognition of receptors.
Introduce our final speaker of the symposium. Stephanie Brocke, who's working in Adrian Rotter's lab, together with Andrew, and will be telling us about cell patterning and developing Arabidopsis.
Good afternoon, everybody. Thank you all for being here, and thank you, George, for the introduction. I know it's been a long day, but we're finally, this is the last talk, so. Okay, so today I'm going to be talking about differential cell growth in Arabidopsis fruit and its relation to the gene fruitful. So what is Arabidopsis and why do we care about it? Arabidopsis thaliana is a model plant, and we care about it in part because it's closely related to the crop canola. And being so closely related, there are many physical similarities between Arabidopsis and canola. These two images here are Arabidopsis, and the three at the bottom are canola. And namely, you can see that there's a very striking similarity between the fruits of the two plants. And these long, slender fruits of this family are termed silics. And it's really important to understand the development and the growth of these fruits in depth, because canola oil is extracted from the black seeds that are found within the fruits of the canola plant. And so we can use Arabidopsis as a model to help us understand the development of these fruits. So in thinking about Arabidopsis fruit development, we're starting with this ovary, which is in the center of the flower. And then in the middle, you see kind of a cartoon image of the silic, which I'll point out the two valve regions on the side are of particular interest. And on the right side of that image, this is kind of just a cross-section of the silic, and you can see the two ovules inside, which will become seeds after fertilization. And then on the right here, this is a depiction of the fruit development, the stages of fruit development of Arabidopsis. So you start with, so stage 14 is right after the fertilization event. And then you see this very dramatic period of expansion and growth of the silics until it reaches about stage 17, which is kind of its fully elongated and fully sized form. So as this very dramatic organ growth is happening, how are the individual cells actually changing and growing? Well, our collaborator from UC San Diego, Juan Jose Radcol, after doing a lot of scanning electron microscopy with these Arabidopsis silics, noticed that the immature and early stage silics have a very uniform cell size distribution along the apical to basal axis. So the cells at the top, middle, and bottom all look very similar. But after maturation, when the silic is about stage 17, there in fact is a bimodal cell size distribution, where you can see the cells at the top and bottom regions of the silic are actually much smaller than the cells in the middle of the silic. So what might be causing this differential in cell size? Well, in order to investigate this, we use a method called live imaging, and this allows us to actually look at these cells and how they're changing as the silic is maturing from stage 14 to stage 17. So to do live imaging, we take a wild-type plant that has been transformed with a fluorescent marker in the plasma membrane, and then we take the whole thing with the pot and all, and we put it under the confocal microscope, and we use the confocal to take images of the top, middle, and bottom regions of a silic of interest, and then we do this every day for a period of 5 days. And this is what some of the raw data from live imaging looks like. So on the far left here, we're seeing this is a stage 14 silic, so you can see how small the cells are. And then after 5 days, on the far right, this is a stage 17 silic. And I've artificially highlighted one particular cell so that you can really see how dramatic the cellular growth is as the silic matures from 14 to 17. So then how do we actually extrapolate and gain insight from live imaging? Well, we start with the raw three-dimensional confocal image like the ones that I just showed you. And then we use a software called morphographics to segment the cells. And then morphographics allows us to make quantifications of things like cell size, cellular growth rate, and it allows us to create heat maps of those quantities. So the thing that you see on the right, the darker reds correspond to a larger cell size, and then the darker blues correspond to a smaller cell size. So after doing some live imaging of a psyllic, where we again imaged the top, middle, and bottom regions separately, we created a cellular growth rate heat map for each region. And so what we're looking at here is the 4 time intervals for the 5 days of growth. And I'll note that in this case, the redder the hue indicates faster growth, whereas the bluer the hue indicates slower growth. So as you can see in the first time interval with the top portion of the fruit, you can see a lot of blues and greens, which indicates that growth is relatively slow at the beginning. And then growth really intensifies, and then by the last time interval, you still see quite a bit of blue and green, and so the growth has slowed, but there is still some more intense growth going on. Comparing this to the middle portion of the fruit, at the beginning, or the first time interval, you see a lot of orange and yellow, which indicates that the growth rate is relatively fast at the beginning. And then again, it intensifies, and by the last day, you still see quite a bit of yellow and orange, which indicates that growth is still like pretty fast by the end. Now this is in striking contrast to the bottom portion of the fruit, where you see that by the last 2 time intervals, almost all of the cells are blue and green, which indicates that the cellular growth rate has decreased substantially by the last 2 days for the bottom portion of the fruit. And if you remember that's in stark contrast, to the top and the middle portions of the fruit. So here I have graphed the growth rates for the top, middle, and bottom regions. And what's kind of important to notice here is that at the beginning time interval, the middle has the highest growth rate. And at the last interval, the middle also has a higher growth rate. And so the growth rate for the middle portion, it increases slightly and then decreases, but in general, it's pretty constant, it's pretty steady. Whereas for the top and bottom regions, they kind of peak quickly by the second time interval and then drop off really quickly. So what we're actually seeing here is that the reason that these cells in the middle are larger is because the temporal period of growth is actually slightly longer for the middle region. So now I'm going to transition into talking about a gene which quickly became of interest and significance to our project. So fruitful or full is a MADS gene, and it is known to be important for cell type patterning in developing psyllics, specifically valve cell patterning. So Juan Jose, our collaborator, created several mutant lines of full, including a weak allele, which is full 6, and then a null allele, which is full 2, as well as an overexpression line. And comparing these three, the genotypes of these 3 lines to the wild type, you can see what a dramatic effect this gene has upon proper psyllic development and proper psyllic expansion. So the mutant lines actually, without full, the psyllic does not expand to its normal length. And so he did a little bit more digging and used scanning electron microscopy to see how are the cells then affected in these mutant lines. And indeed, the cells in the 2 mutants are actually much smaller than the cells in the wild type psyllics. So clearly full is having a very dramatic impact not only upon the organ size, but also upon cell size. So how might full be causing or contributing to the cell size differential that we're seeing? Well, the first hypothesis was that perhaps full impacts a process known as endoreduplication. So endoreduplication is something that can happen when a cell, after duplicating its genome in the S phase, instead of dividing in the normal mitotic cycle into 2 daughter cells, it enters something called the endocycle, where the cell basically will double in size, but it doesn't actually ever split into 2 daughter cells. So what can happen is you can start with, let's say, some diploid cells, and then after a period of division, some of these cells may endoreduplicate, which means that they could eventually end up with 16C, 8C, or 4C fluidity levels, and are correspondingly double, triple, or quadruple the size of a diploid cell. So we wondered, is endoreduplication happening as the cells are expanding and growing from stage 14 to stage 17 is that is endoreduplication kind of contributing to that very dramatic growth that we're seeing. So we use something called a method called flow cytometry, which allows us to actually quantify the nuclear DNA content and the ploidy levels. And so we found that in wild-type stage 14 silics, there's actually a very, very high percentage of the nuclei are 2C or diploid. And there's a very small percentage of octoploid and almost little to no 16C nuclei. But then after doing flow cytometry with a wild-type stage 17 silic, you see that there's a much lower percentage of diploid nuclei and then a much greater percentage of 8C and even some 16C nuclei, which indicates that endoreduplication is indeed happening as these cells are maturing from stage 14 to stage 17, and thus endoreduplication is contributing to the cellular growth that is very obvious that we're seeing. So we wondered, is full having an impact on this process of endoreduplication? So to investigate this, we again did a ploidy comparison between the wild-type silics and the two full mutants. And this is the results of that. And so namely, you don't really see much of a difference between the percentage of 8C and 16C nuclei. They all are really pretty close to each other. And you might expect that because the cells of full 2 and full 6 mutants are much smaller than the wild-type cells, that there would be lower levels of endoreduplication, but we're not really seeing that pattern. So full must not be regulating cell size through endoreduplication because we're not seeing any kind of obvious pattern here. To conclude, cells at the middle region of the silic are larger than cells at the top and bottom regions. Cells at the middle region of the silic grow for a longer time period than cells at the bottom and top. And full affects cell size, although not by affecting endoreduplication. So some future directions would be to do transformation of full mutants with plasma membrane marker to allow for live imaging and cell size quantification, and that will allow us to better understand how the cells of these mutants are growing. And also to investigate the potential involvement of other genes. I would like to acknowledge my mentor, Mingy Wan, Dr. Roeder, for allowing us to be in her lab, or allowing me to be in her lab, sorry. Juan Jose, our collaborator, and all the other members of the Roeder lab, as well as Boyce Thompson Institute and Cornell University in Minnesota. Thank you. Can I take questions?
Yeah, thanks for a great talk. I'm just curious about the overexpression you showed. It looked like there was some curvature going on. Is that correct?
Yeah, it seems so. Yeah, it seems like not only is the silic a bit longer, but it also displays more of a curved phenotype.
And do you know if there's a connection to the patterning of cell size, or?
We haven't really investigated too much into the overexpression line. Any other questions? Yes.
How did you do the flow cell cytometry?
Yeah. So there's actually a machine that is. So okay. So you extract the DNA essentially from samples, and then there's actually a machine called the flow cytometer, and so you just put your sample in there, and then it spits out these graphs that show kind of spikes where there's a high percentage of particles of that size. And so we know, based on other experimentation, that where these certain peaks are, that like those peaks correspond to diploid, tetraploid, octoploid, etc. nuclei.
No further questions? Well, let's thank all the speakers. Students who participate in the summer program, let's please assemble outside and have a sign for the groups.
Vault. Oh wow, I just ate some the camera. Good job, Lily.
I'm hoping one of the other judges has a really strong opinion. Yes, I agree. That's good.